Okay. Generally, we'll start from the question that I did not ask to you because from the topic that I didn't ask you because it's very simple. So the cytoplasm. Cytoplasm is simple concept. It is just the fluid inside the cell. Yes. So we have some extracellular fluid. We'll talk more about this. And we have some intracellular fluid. This intracellular fluid, so the fluid in which all of the other organelles are floating, is the cytoplasm. In the cytoplasm, we have a lot of compounds. So, for example, we have some glucose, some electrolytes, and a lot of very important and very varied proteins. In the cytoplasm, we also have extremely important process called glycolysis. That is, we'll talk more about this later, but generally it's a process of acquiring energy without oxygen. Yes, we are mainly acquiring energy in the form of the ATP using oxygen and using our mitochondria. But we are also able to acquire some energy using glycolysis without using any oxygen. Okay, but first of all, I was asking about the endoplasmic reticulum and Golgi apparatus. Generally, I see that you have an idea what is it, but just to organize, we have the rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is different from the smooth only by one thing, but very important, that in rough endoplasmic reticulum you have those black dots. Yes, what are those black dots? Those black dots are the ribosomes. Ribosomes are the structures that are able to create proteins. They are extremely important. We already, maybe not. Never mind. They are extremely important because they are able to create the proteins. So endoplasmic reticulum that has attached ribosomes to it has the ability to create the proteins. Yes, because the ribosomes can create. And the smooth cannot. The other things are common. So other things are, for example, synthesis of the lipid substances, modification of the proteins that were created, some detoxification, things like that. So generally, this is like a factory a bit. That is, in case of the rough endoplasmic reticulum, producing, in case of the all of the endoplasmic reticulum, modifying proteins. Also producing, modifying lipids and other substances. But generally, how you need to understand those types of structures? Because endoplasmic reticulum and Golgi apparatus are quite similar. Generally, we have some fluid outside of the cellular membrane, of the membrane of the endoplasmic reticulum and Golgi apparatus, and we have some fluid inside of the Golgi apparatus. Between, we have lipid bilayer. Lipid bilayer, that structure is very similar to the structure of the cellular membrane. So why do we have it? We have it to compartmentalize. Yes. So when you are looking, for example, at this part or this part or this part, these parts are a bit cut out from the other parts of the endoplasmic reticulum. Yes. They are joined. Yes, but the movement is not as easy, and they are completely separated from the cytoplasm. Because of it, there may be completely different chemical reactions happening inside. Yes, and this is a critical idea. So we have generally different chemical reactions that are happening inside the endoplasmic reticulum and the Golgi apparatus than in the cytoplasm. That's why they are created, yes, because they are just the sacs with the enzymes, to be honest. So these are the structures, both endoplasmic reticulum and Golgi apparatus, that are generally modifying some substances, yes? So, what is the difference between the endoplasmic reticulum and Golgi apparatus? Golgi apparatus generally do not have ribosomes, although from every rule there are some misconceptions. Never mind. But generally they do not have. And usually Golgi apparatus is about higher modifications of the substances. So the usual way is that the protein is created in the rough endoplasmic reticulum, then modified, then sent by the vesicles into the Golgi apparatus, and Golgi apparatus is further modifying it before it is sent somewhere. So the substance modified in the Golgi apparatus may be sent outside of the cell, may be sent to some other part of the cell, or maybe just kept in the Golgi apparatus if it's needed there. Yes? Or maybe released just into the cytoplasm without any problem. Okay, thank you.